Hello students, today we are going to discuss about life history of Rhizopus. Okay, life history or you may say life cycle. Whenever we will ask to explain the life history or life cycle, then you have to mention minimum three things. One is their systematic position, another is their thallus structure, and next one is their reproduction. Okay, so this is the systematic position of the fungi Rhizopus. It is included in the kingdom Fungi, division Eumycota, subdivision Zygomycotina, class Mucoraceae, family Mucoraceae, and their genus Rhizopus. Okay, so this is the systematic position or the latest modern systematic position of Rhizopus. Then we are going to discuss about their thallus structure. Okay, this is the minimum thing you have to remember. Nothing is there. Then we are going to discuss about their thallus structure. Okay, thallus structure or you may say vegetative structure. What is their structure? The plant body or the fungal body of Rhizopus is a thallus and it is made up of several hyphae. Okay, it is made up of hyphae. These hyphae are generally three different types, or the hyphae of the Rhizopus is classified into three different parts. Before going to those parts, we have to know that which type of hyphae is this. The hyphae of Rhizopus is eucarpic in nature, eucarpic and cynocytic, C-O-E-N-O, cynocytic in nature. These hyphae are slender tube-like structure. This is the hyphae, slender tube-like structure. These are eucarpic. Eucarpic refers to those thallus or those hyphae where a part of the hyphae is developed into sporal or sporangium, spore producing body or sporangium. Where a part of the thallus is developed into spore bearing body or sporangium, then that is called eucarpic. Then its opposite is holocarpic. Holo means whole. When a total thallus is converted into spore bearing part or sporangium, then that is called holocarpic. Okay, the part, the hyphae of Rhizopus is eucarpic, means a part or a segment or a part of the thallus is converted into sporangium. Then cynocytic. Hyphae of Rhizopus is cynocytic. Cynocytic refers to a single cell in which more than one nuclei are present. Okay, when in a single cell more than one nuclei are present, then that is called cynocytic condition. This cynocytic condition arises due to the more than one karyokinesis and when the cytokinesis is absent. When the cytokinesis is absent and only karyokinesis occurs, then the cell developed is called cynocytic cell, where in one cell more than one nuclei are present. Okay, so the hyphae of Rhizopus is eucarpic and cynocytic in nature. Eucarpic and cynocytic in nature. Then the hyphae of Rhizopus is classified into three different parts. Three parts. Okay, three parts. First one is called rhizard, R-H-I-Z-O-I-D, rhizard. Second one is called stolon, S-T-O-L-O-N, stolon. And third one is called their sporangiophore, S-P-O-R-A-N, sporangiophore, P-H-O-R. Mark here, I am writing sporangiophore. There is another term also, sporangiospore. So there are two different things, sporangiophore and sporangiospore. Sporangiophore is a part of the hyphae, inside which sporangiospores or spores which are developed inside the sporangiophore are called sporangiospores. Okay, see, the part of the hyphae or the hyphae is classified into three different parts. First one is called rhizard. Okay, rhizads are grow inside to the substratum. Okay, these are the rhizads. Rhizads are growing from the bunch or tube-like structure. These are the tube-like or slender structure. Rhizad, tube-like, slender, S-L-E-N-D-E-R, slender structure growing inside to the substratum. They are going inside to the substratum or the host cell. Then the stolon. This is the stolon. It is the stolon. stolon, stolon, or it is a parallelly. This stolon is going parallelly to the substratum. Okay, parallelly going to the substratum, and it is the aerial part. Stolon is the aerial part developing parallel to the substratum. Then sporangiospore. Sporangiospore are the spore producing body like this. They are growing opposite to the rhizode. Sporangiophore. These are the sporangiophore. Sporangiophore are the aerial part. They growing opposite to the rhizode. They bear sporangia. These are the sporangia. This is sporangium. Singular is sporangium. If I mention plural, two more than one, then I will mention this as sporangia. Inside this sporangia, sporangiospores are developed, which are helps in asexual reproduction. So this is the main or simple thallus structure of the Rhizopus. Rhizopus thallus is consist of hyphae. This hyphae is eucarpic and sinusoidal in nature. Then the hyphae of Rhizopus is classified into three different parts. The part which grow inside to the substratum is called a rhizode. It helps in anchorage and absorption. Then stolon is the aerial part. This grow in the parallelly to the substratum. Then sporangiophore. Sporangiophore develop opposite to the rhizode and they bear spores, which helps in asexual reproduction. Okay. So this is the thallus structure of a Rhizopus. Then we will going to discuss what is the type of their reproduction. A reproduction of Rhizopus occurs by two methods. One is asexual method, another is sexual method. Okay. Reproduction of Rhizopus occurs by asexual method and sexual method. First we will going to discuss about asexual method. As I mentioned earlier, the sporangiophore. Inside the sporangiophore, spores are developed, and those spores are called aplanospores. Aplanospores. These aplanospores are type of sporangiospores. These are the sporangiospores which are non-motile. That's why the non-motile spores are called aplanospores. These aplanospores are the asexual reproductive part. When they fall on the substratum or any host cell, then they grew into new thallus structure. Okay. So by means of aplanospores, Rhizopus do their asexual method. So we are going to discuss how these aplanospores are developed. Okay. Development of aplanospore. How this aplanospore is developed? First, see, this is the sporangiospore, this is the stolon, this is the rhizode. Okay, this is the thallus structure of the Rhizopus. This is the sporangiophore, PHR. This is the sporangium, GIUM. First, what's happened in the first stage of development of spore or aplanospore, the sporangia and its protoplasm is classified into two different parts. One is outer, another is central. Outer and central. This is the outer part and this is the central part. Outer sporogenous part and inner columella part. In the next stage, what's happened? In the first stage, the protoplasm is classified into two different parts. Then in the second stage, the outer sporogenous part is classified or cleavage occurs. Cleavage or breaking down. Cleavage occurs to the outer sporogenous part and a multinucleate segments are found. Cleavage to sporogenous part and to form multinucleate segments are found. Then in the next third stage, these multinucleate segments are covered by a protecting layer, and These protecting layered structure, multinucleated structure, are called nothing but your aplanospores. Okay. Then in the fourth stage, the central columella is grow inverted. Okay. In the fourth stage, columella grow inverted basin-like structure. Inverted basin-like structure. That means like this. Okay. This is the inverted basin-like structure. The columella grow. This is the central part. Columella. This is the outer part. Columella grow into central inverted basin-like structure. These are the aplanospores. Due to this inverted basin-like structure growth, the sporangium wall breaks, and these aplanospores are exposed to air, and dehiscence occurs. When these aplanospores after dehiscence fall on any substratum, then they grow into your new thallus body. Okay, this is the asexual reproduction process. Then we will going to discuss about what is the sexual reproduction process. Sexual reproduction process. Sexual reproduction process is occurs by gametangial copulation. Occurs by process gametangial copulation. In gametangial copulation process, two different strains of hyphae. One is positive, attributed as positive, another is attributed as negative. Two different strains of hyphae came into contact with each other. Okay, these different strains are morphologically distinct but morphologically similar, but physiologically they are dissimilar. Okay, morphologically means by their structure and their properties. Morphology, outer structure. Outer structure, in the outer structure or outer view they are similar, but physiologically they are different. So two different strains hyphae are first came into contact with each other due to a hormone, then hormone called trichoric acid. Okay, that is not important. After their contact, the part which contact or the tip of the tip is called this part. This growing part is called your progametangia. Progametangia. The tip of the progametangia is called gametangia, and the bottom part is called your substratum. This is the bottom part. This is the gametangia. This bottom part is called your substratum. Suspensor. Bottom part is called suspensor, and the tip is called your gametangia. Okay. Progametangia. The progametangia consists of two different parts. The tip which came into contact with each other called gametangia, and through which it is attached to the main hyphae is called your suspensor. Okay. Next, after the attachment of gametangia, development occurs. Then development occurs to the gametangia, and they develop into development occurs. Okay, this is the developed structure of a gametangia. After the development of a sporangia, gametangia, after the development of a gametangia, the wall between these two gametangia are ruptured. Okay, then the protoplasm and the nuclei of two gametangia intermixed with each other, and after this intermixing of the protoplasm and nuclei, the structure found is called zygote. This is the structure found after their intermixing of protoplasm. This structure is called zygote. This is the diploid stage of the Rhizopus. Till now, whatever we discussed, that is the haploid stage. This is the first diploid stage called zygote. This zygote undergoes development to form zygospore. Okay, this zygote undergo development to form zygospore. This is called zygospore. This zygospore consists of an outer wall and an inner wall. Then, when favorable condition occurs, this zygospore converted into or grows into germ cell. You may say germ cell. This germ cell, when favorable condition arise, detach from the hyphae and fall on the substratum, then grows into a new thallus. Okay, so this is the sexual reproduction. Sexual reproduction occurs by gametangial copulation. In the gametangial copulation, progametangia participate. When the two different strands came into contact with each other, their tip or the gametangia contact with each other, then the tip part undergoes development and their wall is vanished. When the wall is vanished or interrupted, then the protoplasm and nuclei of two gametangia intermixed or intermingled with each other. After that, the structure is formed. It's called zygote. This zygote develops into zygospore. This zygospore, under favorable condition, grows into germ cell, and this germ cell, when detached and fall on the substratum, they grow into a new thallus or the rhizopus. Okay. So this is the general life history of Rhizopus. It is very simple, and if you want to mention their nature and occurrence, this is the simple description of the Rhizopus. You may include this before the vegetative structure. Okay. After the classification, systematic position. After the systematic position or classification, and before the thallus structure, you may mention the nature and occurrence. Rhizopus, nature of the Rhizopus is a parasitic fungi. Okay, that's why it grow on the other substratum. And occurrence, where it is found, it is generally called bread mould. B-R-E-A-D, bread mould, M-O-U-L-D, bread mould. It is found in bread or in any fruit or vegetables which are kept for many days in home. Then after in those kept food particles, some fungi are developed. Those fungi are generally white or brown in color. Those are generally called Rhizopus. Okay, these are also generally called bread mold, found in the household fruit bodies like your bread or your fruit vegetables when kept for many days. So this is the nature and occurrence. If you want to mention this, then mention before the thallus structure and after the systematic position. Okay, first systematic position, then nature and occurrence, then their thallus structure, then their reproduction. Reproduction is sexual type and asexual type. Asexual and sexual type. Then the things arise. Life cycle. What is the type of life cycle of Rhizopus? Type of life cycle. Type of life cycle is, see, zygote is the second diploid stage. Before zygote, all hyphae, all thallus structure is your generally haploid in nature. That's why the life cycle of Rhizopus is called haplotic. Haplontic type. Okay. Haplontic type, because the haploid stage is dominant. Only if sexual reproduction occurs, then zygote is found, then your diploid stage is found. But if asexual reproduction occurs and all the zygotic phase, or hypha phase, your plant body or the fungal body of Rhizopus is haploid. That's why the life cycle of Rhizopus is called haplontic type. Okay. So this much you have to write if the question arises, life cycle of Rhizopus. Okay. So this much in this class. We will discuss further in the next class. Okay. Thank you.